Hello, everyone. Welcome to Mind Over Matter, a podcast run by Youth for Neuro. I'm Anika, a rising senior in high school and the host of today's episode. Today, I'm joined by Dr. Paula Desplats, a professor at the Departments of Neuroscience and Pathology at UCSD. She received her PhD from the University of Mar del Plata in Argentina and completed her postdoc at the Scripps Research Institute, researching Huntington's disease, and later joined UCSD, where she extended her training in Alzheimer's and Parkinson's diseases. Her lab focuses on the role of epigenetic mechanisms in neurodegenerative diseases, particularly Parkinson's and Alzheimer's. We're glad to have you today, Dr. Desplats.
Hi, Anika. I'm very happy to be here to share our research in this area.
All right, so to get started, what is neuroepigenetics and what led you to researching the role of epigenetic mechanisms in neurodegeneration?
Okay, so that's a very, very deep question or large question, but we can start by thinking in epigenetics by the very basic concept. And the word epigenetics, epi means above, describes all those modifications that won't be involved in changing the sequence of letters in the DNA or the pure genetic content, but still can modulate the genes and how they express. So if you think in context, all of the cells in our body share the same genetic code. We have the same DNA, same sequence everywhere. Yet some cells will specialize in becoming skin cells, others will be hair, and others will be brain or heart cells. So how those cells become their specific fate and turn in certain specialized structures is dictated by epigenetics. So if you imagine that the whole information of our genome is in a book, epigenetics would be like putting a bookmark in the pages that had to be read in each of these cells. So these, as you can imagine, these mechanisms are very important for cell functioning and determining what has to be expressed and used at certain times. So it has a major involvement in development. So during development, these epigenetic mechanisms are very active and remodel and dictate how the cells will be developing, dividing, and forming different organs. So it was not until recently, in certain ways, let's say 2 decades or 3, that we started to understand what was the role of these epigenetic mechanisms in the brain, because when we talk about cell division, we don't think in neurons, because neurons are post-mitotic and don't divide during our entire life. So during many years, it was kind of certain skeptical thinking about how does DNA methylation or histone modifications that are actually the mechanisms of epigenetics had to do with the brain. And fast forward many years of research, we found that these mechanisms are very important for the plasticity of the cells. And for example, we now know that the histones need to change and be modified as we record our memories or as we recall our memories. Kind of with years, we understood that these roles, these mechanisms have a very important role also in brain functioning. And this field of neuroepigenetics was born. And basically what we studied is exactly those functions that make this DNA changing expression in brain cells. And as you know, the brain is one of the most complex organs. We have different cell types, and we have these cell types specializing in different regions. So it's very complex also to get to approach studying these mechanisms. And we have learned with the advance of technology many details in how rich these modifications are. And basically, when we think in epigenetics, we broadly talk about 2 mechanisms: histone modifications. Histones are proteins, the proteins that form a core where the DNA is wrapped around, and they have some tails that protrude from that coiling that can be modified. And these modifications come in many different flavors. And depending on these modifications is the type of structural function that we will uncover. The other broad modification is DNA methylation, and this is the inclusion of methyl groups into certain specific sites on the DNA. And when we take the balance of both histone modifications and DNA methylations, it's like imagine that you have written a message in a paper and now you start folding it very tightly. While the paper is folded, you cannot access the message. So at certain moments, if you need to go back and read and remember that information, you will unfold this and that information will be accessible. And that's kind of in a nutshell what these mechanisms do. They change the way the chromatin is folded to enable the access to genetic information and permit transcription of gene information, or it really blocks by doing this high order of folding and chromatin state that will really not let the genes be transcribed. So this is a balance that happens in the cells, and it's quite more dynamic that we expected before to encounter. And these are the functions that are important for neuroepigenetic as well. So it can respond to certain states in the cells or in the body. It can help fundamental functions. And another very specific things about epigenetics is that these mechanisms are very linked in responding to our environment. Being environment broadly understood, like our bodies being exposed to certain environmental pollutants, for example, or environments, even light, or talking about the cell environment and what happens inside our bodies and how those cells can be modulated by responding to inflammation and to disease states or to different nutrients.
Right, yeah, okay. So you mentioned DNA methylation as an important epigenetic mechanism. So how exactly does that contribute to like neurodegeneration?
So that's what we study, basically, right? And when I started this road out after my postdoc and trying to define the topic or the theme of my own lab, I became very interested in epigenetics because it was kind of these neuroepigenetic fields, it was starting to flourish and emerging. And I wonder how that could be a part of neurodegeneration mechanisms. So in the umbrella of neurodegenerative diseases, we have many disorders, and these disorders are characterized by the accumulation of proteins that are not folded properly and form disaggregates. In Alzheimer's, it's the senile plaques. In Parkinson's disease, we call them Lewy body. But these proteins disrupt the cells in many ways. Not only they stop functioning, but also they can pull other proteins, so these are no longer available to fulfill their roles. So my question was, has DNA methylation anything to do, since I was very interested in understanding changes in gene expression that happens during disease? So my first study in this field was really trying to see if there were changes in DNA methylation in the Parkinson's brain. And we had mouse models of disease, we have cellular models of disease, but also we have postmortem samples from patients. And what we observed is that it was a decay. There was less DNA methylation overlap in the DNA associated with Parkinson's. So when we started to understand the mechanisms and knowing that alpha-synuclein is a protein that starts or is the center of the pathological findings that we found in the brains, we started to see what potential interactions. And we found that alpha-synuclein can bind and pull off a very important enzyme, that is the DNA methyltransferase. That is an enzyme that can take these methyl groups and put it in the DNA. So by having these trapped into the cytoplasm, they cannot reach the DNA, and potentially that causes a decay in DNA methylation. So that is only one of the potential contributions. But after that, many other groups found differences in DNA methylation in other disorders. And for example, working in Alzheimer's disease, we also found many changes in these brains, and that the changes seems to be specific of certain disease stages. So then we started to understand that these have a larger impact on how these genes can be expressed and potentially can even contribute to augment the neuronal decay or the neurodegeneration process itself.
Okay, so like as you observe different stages of neurodegenerative diseases, how do you see that DNA methylation and like histone modification changes?
So as I mentioned, there are different type of changes. In terms of histones, there are multiple changes that happen, and there are decays in very specific histones and very specific groups. So there are more broad, very hard to describe as a list, but there are a lot of rearrangements happening that are associated with which portions of the genome get expressed. And we found, for example, changes that favor that pro-inflammatory genes become expressed. So that's part of the disease pathology. In terms of DNA methylation, similarly, we and others found, for example, in Alzheimer's brains, that there are changes in pro-inflammatory genes at the earliest stages, so that might contribute to disease progression. And it's it's a genome-wide change in this DNA methylation. Another very interesting thing that happens is that there is a normal pattern that we call epigenetic drift as we age. So as we age, our cells start changing these patterns of DNA methylation. Some places get more methylation, other places in the genome get fewer methylation sites. So this is so specific that you can trace the methylation levels of certain particular marks throughout the genome and create what is called the epigenetic clock. You can predict the age of an individual very, very specifically. So after many organisms and cells have been measured, there is a known pattern of epigenetic changes, which we consider normal epigenetic aging, because it really correlates with the biological aging. If we plot now the changes in someone that has a disease, and that slope change, we can calculate that there is an accelerated of epigenetic aging. And this happens in Alzheimer's disease and other disorders as well. So we know that there are enough changes in DNA methylation that alter the way an Alzheimer's patient is aging in terms of cellular events.
Okay, does that also help in like detecting like which patients might be at increased risk for diseases such as Alzheimer's?
Not as a predictive marker. So this is kind of an understanding of how the disease is progressing. But one of the interests in my lab is trying to use DNA methylation as a predictor or a biomarker. And why are we interested in biomarkers? Well, these disorders happen in the brain, so we cannot take samples as when you get a biopsy in cancer. So we need what we call proxy tissues. And the easiest will be a blood sample, for example. So we have been investigated in the Parkinson's group whether we can find different signals of DNA methylation in the blood from patients comparing to healthy adults of the same age. So we have been analyzing thousands of samples with the support of the Michael J. Fox Foundation. And we found that there are changes in these patterns. So we are working still in trying to identify how good are these marks that are different in predicting or in being very specific to tell that a person has Parkinson's disease. We found other interactions, for example, people that are already taking medications for Parkinson's have a different profile of DNA methylation. So maybe we could find certain things or certain marks in the genome that can help us understand how these patients respond to medication, to existing medication, and potentially a new one. So that's one of the things that we are doing.
Okay, nice. So, as I understand, there are many factors that like are relevant to epigenetics, like environmental factors. So, what factors do you notice contribute like the most to neurodegeneration?
So that's not exactly what we study in the lab. We know that, for example, in the case of Alzheimer's, um, there are not many genetic mutations, but there are some changes in the genome that we call variants that will not alter the genes, but have a variation, a slight variation of the function that can predispose an individual or increase the risk. But there are other that are metabolic, for example. And if you have certain diseases that we call comorbidities, you are at higher risk to develop Alzheimer's. Like, for example, if you have diabetes, if you have hypertension, cholesterol imbalance. In terms of environment, there is good documentation of that exposure to lead seems to be associated with increased risk for Alzheimer's. And this is honestly a new field. So there is a concept that is being pushed now that is the exposome, and trying to exactly do that, define what are these things to which we are exposed with new knowledge that we have now, right? So even microplastics or BPA or other things that could have had an impact. But that's really an emerging field. So now we have more tools, we have more capacity to find these marks in the DNA, and we can do prospective studies and look for people as they develop the disease. So I think that knowledge is coming in future years.
Right. So, looking through your recent publications, I saw that you investigated circadian rhythm disruptions, particularly in Alzheimer's. So, what can this tell us about Alzheimer's pathology?
Okay, so we can see in circadian disruption also as being some part of the exposome. So let's recap a little bit and say a few words about the circadian system. And the circadian clock is a system that it's very well preserved across all the phyla and all the organisms. And that is because we need to adapt to live in these planets that rotate and expose us to periods of light and darkness. So our bodies need to be ready at all the levels. If you are a plant, you need to be ready to photosynthesize when the sun is out. If you are an animal, you need to be ready to feed, to do your activities, reproduce, move around different environments, find shelter during night. So this is a very exquisite system that in fact is built upon gene expression regulation. And the way this works at the molecular level, so for example, in terms of environment, we receive the input of the light through the retina, and that will be transmitted using ganglion cells, very specific with specific functions, into the central biological clock that is in the hypothalamus, that is called the suprachiasmatic nuclei. And this is a very tiny region that have these cells that will transduce this environmental input into signals in gene expression. And the way the clock do the rounds is just by expressing different genes at different time points. So what we call the core clock has certain genes that will be expressed at a certain moment, and they will also activate their own repressors. So when the repressors are up, these other first genes will be down, and this will produce a sinusoidal curve, and it's up and down every 24 hours. But this core clock also will send the signals to many, many other genes in the body. Depending the organ, 40% of the genes can take these rhythms of expression. So I think recently we have been also starting to realize how important it is that we are synchronized with our environment. And the way, unfortunately, that we realize that is because we now are exposed to very disrupted signals. We have access to food 24/7. We have access to light 24/7. This artificial light, when we are exposed to, even in our phones, in the TV, in computers, it's also very pervasive. And the signal of this different light illumination that is way more strong than the light from the sun can last hours in our brain. So we know, for example, the many issues in health that the shift workers happen to experience, right? So all these nurses or firefighters that need to be up during the night, really suffer this adjustment to the body. And they have deleterious effects throughout. So it happens that patients with Alzheimer's per se experience what we call circadian dysfunction, and they express that clinically by being more agitated, for example, during the evening, at certain moment of the day when the sun goes down. This effect is called sundowning. The patients get more agitated, confused, not remembering people or names, and felt threatened. And the other thing that happens is the change in the way they sleep. Sleep cycles are controlled by the clock. And these patients also shows that they are very sleepy during the day, taking several naps, and then at night, they are very active and agitated at different hours. So these two factors are the most important contributors for these patients to require being placed in a nursing home, because really the family cannot cope with these events. But they also affect the way their bodies work, and they also affect cognitive skills. So we were very interested because 80% of the patients suffer these circadian alterations. And in the beginning, it was assumed that this was a consequence of neurodegeneration happening in the brain. But as we collect more data, many groups have shown, studying even behavior in people, that this circadian disruption happens many years before the actual neurodegeneration or even the big memory complaints. So these are early events. So from my lab, studying DNA methylation, there were a couple of studies showing that DNA methylation was important in mouse, for example, to adapt to changing light cycles. So we had all this data from our studies in postmortem human samples. So we went back to that, and by using the time of death as a point of time in the day, we found that DNA methylation was also oscillating. So we had our first evidence that these epigenetic mechanisms were part of this problem in the dysfunction of the circadian cycles in patients, because we found that these marks in methylation were very different in the patients than they were in healthy donors. And in fact, these were even worse in patients that were transitioning the earliest stages of disease. So that's how we became very interested in this study. So we took these animal models and decided to map what are the pathways or the genes that are controlled by the clock in the brain. And we focus in the areas that are more affected by Alzheimer's, which are the hippocampus that controls memory and the cortex that controls our social and behavior interactions. And we mapped that many of the genes in those tissues are rhythmic, so follow the clock. But very importantly, we found that many genes that we know are associated with Alzheimer's also have rhythmicity. So when we went to our animal model, we found that these rhythms were very disrupted. Many of these genes stop being rhythmic, that are flat throughout the day, or they are peaking or being expressed at times that are not the correct ones. So finding this disruption also kind of created a way or the idea of, can we do something for these animals? And can we try to fix the clock somehow to get these animals to be better?
Okay, that's really fascinating. So, now I also saw that you used spatial transcriptomics in your research. Could you describe what that is and how it relates to your research?
So this is a very, very new technique. Usually when we want to see how the genes are expressed, we take a little piece of tissue and we isolate the mRNA, the messenger RNA, that is how the genes are transcribed and expressed. And that is called bulk tissue, and we need to grab a little piece, right? So once we isolate that, we just get a portion of the brain and we mash everything together and we know what is being expressed, but not necessarily where. And yes, we can be very fine and dissecting. So we can say, oh, it's in the hippocampus or it's in the portion, but that physical dissection, imagine a mouse brain is very tiny, has its limits, right? So you could see what, but not where. So this is what the spatial transcriptomics does. This is a new technology where you grab like a slice of the brain, and instead of smashing and getting the RNA out, you first add what we call a tag molecularly. And that tag is like if you were just adding a zip code. And then you grab all the RNA and you do sequencing. So you know what are the genes that are being expressed, but now you go back to your data and you find the zip code. So you can see from one slice of the brain, the genes that were expressing many different regions. And since these regions are functional, we can start building networks and understanding how what was thinking not only in the hippocampus, but in the sub-region of the hippocampus, and how that relates to something in the cortex, but not just in the cortex, but in one of the, for example, 6 layers of different cells in the cortex, and maybe only in the posterior part of the cortex versus the anterior. So you get this amazing resolution of the entire brain in terms of mouse that enables this, what we call system biology, and understanding how these networks interact, because the genes don't express just by themselves. It's always a network that will kind of transduce a function to the cell. So it has been a huge advance in the way of we can understand how these networks work.
Nice. Okay, so that's seems like a pretty important technology that you're using. So, what else is what other types of technology do you use that is essential to your lab's research?
We use different types of ways of understanding gene expression. So we talk about epigenetics, right? So that's the methylation levels in the DNA. So to measure that, we do what is called bisulfate sequencing. And what we do is we need to do a reaction with this bisulfate acid into the DNA. So we extract from the cells the DNA, and we do this assay that puts like a different mark where a methyl group was. So then when we sequence and we know all the letters and the DNA and the code, we can have this little mark that we know, oh, a methyl group was exactly here. So then we can measure DNA methylation. Another thing that we use is called attack-seq or sequencing, and that's basically a way of measuring those states of opening of chromatin that I mentioned before. So we can measure where the chromatin expanded to enable gene expression. So then we can assume that then those genes are being activated. And we can start finding what are the sequence that kind of enable this? What are the regulatory regions? What are the potential factors that can be helping this opening or the closing? And another one, the one that we use most, is RNA-seq. And as I mentioned before, is sequencing these pieces of RNA that is how the genes are transcribed into proteins. So we can sequence that RNA. We extract the RNA and we sequence. So in the past years, we have been seeing these emerging technologies like the spatial, as you mentioned, but also we have now more and more capacity of doing this sequencing at a different level. It means that we have better resolution. And there are new techniques that also are single cell, because I mentioned in the beginning, the brain, it's very specialized. You have different type of cells. Each cell type, neurons, microglia, oligodendroglia, myelinating cells, have their own epigenetic and genetic code, right? So we need to understand those events of gene expression. So with single cell, you can take a piece of tissue and really separate these cells and analyze them very specifically. So that's a resolution that before we didn't have. So we are learning a lot as a field, I mean, in terms of how all these mechanisms are important in the brain when it's healthy and the basic functions, and then when disease situations arise.
Yeah, so you talk about how its technology's already helped a lot in your research. So how do you hope it will advance research in the future?
Well, as I mentioned, by having these more precise maps of what is being expressed, we can start finding what are the mechanisms or what are the key points here and there that can be targets. So the thing that we are failing and missing is having a cure for these diseases. And very recently, you have our first potential treatment for Alzheimer's approved after 20 years, right? So, and so we need more. So if we identify how these things are managed and regulated, we might think that potentially we found a target that we can produce or create a drug to tackle, to normalize and see if we can prevent this cascade of neurodegeneration. And the other area is biomarkers. By having more resolution to screen not only gene expression, but also now the proteome. What are the proteins that are being kind of synthesized and at play at certain moments and how they vary in disease? We can also potentially get better markers that we can use to diagnose and predict the disease. The earlier we can diagnose a patient, the better our chances to really modify or treat disease. Because unfortunately, when these disorders manifest clinically, let's say Parkinson's, when the patient have these tremors or motor impairments, or Alzheimer's, when the patient have this memory impairment, the neurons in the brain have been lost already, and we yet cannot replace those neurons. So the sooner we can understand this process is happening, as I mentioned before, we cannot access the brain, but we know that these processes happen many, many years before this clinical manifestation, maybe more than 10. So if we can start recognizing very subtle changes and we can start testing and we can just detect these patients early enough, we at least might change the way the disease progresses. So if we can attenuate the progression of disease, we can have way better chances and outcome for the patients.
Yeah, it sounds wonderful. So, in terms of your research, what are you most excited and hopeful for?
Okay, so what I'm more excited is that my lab, that it's basically very deep molecular research, it's now entering in doing a first clinical research with patients in the terms of circadian dysfunction by working together with the Alzheimer's Disease Research Center here in UCSD. And that's a huge translational event for us. So what I'm excited is like now by having this collaboration, these multidisciplinary teams where the clinical enterprise is joining the molecular research, we can really start talking to each other in terms of specialization and reaching the patients in a more faster and efficient way. So with all these new tools, we are learning a lot of information and we are understanding disease at a different level. And I think that's exactly what we need to crack these diseases and really try to find a cure for patients. So I'm excited to potentially see that.
Oh, yeah, sounds wonderful. So yeah, thank you for joining us today, Dr. Desplats. Um,
Thank you very much for the invitation and the opportunity. I think it's great that we can share what we do in the lab and hopefully inspire new careers and people that want to join this type of research and really dedicate. We really need more people to fight these diseases.
yes, yes, of course. So for our listeners, be sure to check out Dr. Desplats Lab website linked in the episode description to see more of her work and publications. If you enjoyed this episode, be sure to give us a follow at Youth for Neuro on Instagram, Spotify, LinkedIn, and also be sure to check out our website linked in our bio. Until next time, this is Anika signing off.